Are you able to see my presentation?
Yes.
Okay, and hang on. How do I take this down? Go away. Go away. Can I not hide this bar, Munazim? Hide floating meeting controls. This is the video panel, I think. No, not that. Sorry. I don't want to hide video panel. Show video
We
panel.
can see your PPT, ma'am. It's showing.
Yeah. No, I don't like the floating business. Yeah, I've taken it out. Okay, people, if you're ready, I can get started. Renita, you can hear me still.
Yes.
Awesome. Great.
Let's start.
Okay, hello everyone. Thank you for joining me online today despite the patchy internet. I'm here to sort of tell you people a little bit about why analog research is important. And I'm going to come from a place of trying to give you a sense of how we use astrobiology analog research to understand how life might have come about on the early Earth. So there is going to be a little bit of introduction that you need to have background sort of context before I can jump into the sort of more detailed part. I'm still going to keep it very sort of broad. I'm not sure about the background of the diverse participants, so I'm just going to keep it minimally sort of jargony. But if there is anything that you have to ask me, internet connection permitting, we can have a conversation. But I'm going to start now. So we try and understand how life originated on the early Earth. And in order to assemble the most minimal of life in the lab, in addition to studying lab simulated experiments, it is extremely important to go out and do analog research or bring back samples from analog field sites in order to better understand how this transition from non-life to life would have happened on the early Earth, or how the transition from chemistry to biology would have happened on the early Earth. And quick acknowledgements to say that nothing would be possible without the amazing bunch of students that have gone through my lab and are still in my lab, a whole bunch of research scholars, a postdoc, and undergrads who make this research journey very exciting for us. We call ourselves the Chemical Origins of Life Lab because that is precisely what we try and understand. How did life chemically originate on the early Earth? So when do we think this transition happened? Here is the early timeline. Sorry. I don't know if somebody said something. I'm going to keep going. Here is an early history of life on Earth. And this kind of gives you a sense of the timeline that we're looking at, where we think these events would have panned out. We're looking at events that happened somewhere between 3.8 to 4 billion years ago. Very early on in the history of life, once it attained a stable hydrosphere, which we are not very sure when it happened, but it was super early after its formation. Very interesting prebiotic chemistry set the stage for the availability of lots of interesting precursor molecules that are relevant to life's biochemistry. But how did you go from those prebiotic soup of molecules to a space where we can recognize the molecules much more readily, like we see in life biochemistry? And some of these molecules that I'm referring to actually are your nucleic acids, your proteins, your lipids, your carbohydrates, so on and so forth. These are the four sort of major classes of biomolecules that we see in life today. So how do you go from that prebiotic landscape of precursor molecules to what it is today is something that we do not know how it exactly panned out. There are systematic ways in which you can think about it, but you need to have a lot of information about the early history, geological history of Earth. You need to have a good bit of understanding about the environment that would have been present on the early Earth, or you have to make some reasonable approximations of it in order to understand these kinds of important transitions that would eventually allow us to understand what are the processes, what are the niches, what are the pathways that allowed for the emergence of life on the early Earth. So when I keep saying emergence of life, we need to have some working definition in order to be able to systematically approach this in the lab. And for this, there was a working group that was constituted in the early 90s, and they came up with this definition, which works very well. It basically addresses life as a self-sustaining chemical system that is capable of Darwinian evolution. And the hallmarks, important hallmarks of life include these three essential features. One is you need to have a molecule that encodes information, and that in our cells are nucleic acids, predominantly DNA. DNA is what encodes information in modern cells, and RNA helps in transcribing this information to make a lot of important catalytic molecules called proteins, which essentially run the cells, basically, which power the various reactions of life. So self-replicating molecules, super important. Metabolic network of interacting molecules, very, very important, because life is a far from equilibrium process, and for it to be able to sustain itself, you need a bunch of molecules coming together to give some sort of an energy output. And finally, if you look at all of life, it's a compartmentalized system. So a compartmentalization sort of a scenario would have been crucial to minimal living entities. So in this context, sorry about that. Oops, see what is happening. Yeah. How can we think about this transition then? Why is it called the chemical origins of life? I mean, this is encapsulated in the slide, which shows you the continuum that exists between physics, chemistry, and biology. If you look at these biological molecules here, let me try and see if I can get a highlighter. No, that's not a good idea. Laser pointer is better. So if you can look at the biological molecules here, I've represented two molecules, one a proteinaceous enzyme called cytochrome C. Another one is a very important enzymatic molecule, but not a protein molecule. It's an RNA molecule, but nevertheless catalyzes reactions. Zoom into them, and what do you see? You actually see that they're made up of simpler monomeric molecules, which are in the chemical realm. Zoom further into them, what you see is they are fundamentally made up of a whole bunch of atoms. So all of life molecules are made up of C, H, N, O, P, and S atoms. Yeah, carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Predominantly, you do have contributions coming from a lot of different kinds of metals, but that is secondary. Fundamentally speaking, organic life has all these atoms in it. So if you can think about this beautiful continuum, you can actually start systematically addressing the question of transition from non-life to life, to the transition from chemistry to biology. Are you guys still with me, Pranita?
Yes, yes.
Awesome. Okay. So the precursor molecules, how did they come about on Earth? And this is where analog environments become very important. There are two hypotheses here. One is that they were delivered via exogenous processes, basically meteorites, comets that kept striking the Earth very early on in its history. And the proof for this is all the impact craters present on the moon. So there's a thought that about 3.9 to 4 billion years ago, there was a lot of heavy bombardment that happened, which brought in a lot of these interesting molecules, which can also be readily formed in the interstellar medium. Another important hypothesis posits that processes that occurred in terrestrial geothermal niches on Earth, like the ones that you are going to see in Ladakh at some point, these processes also allowed for the population of molecules in these environments, like the one that is shown on the right panel here. So either via exogenous delivery or via terrestrial synthesis, you could have potentially had the monomeric molecules, which then came together, had a lot of interesting interactions, eventually leading to the formation of living entities. So what do we know thus far? In the time since when this field of research started in 1953, the same year as DNA structure was explained, up to now, we have a reasonable sense of how from the inorganic prebiotic soup can you make the precursor molecules relevant to life. But how do you go from those precursor molecules to more complex biopolymers? Because look at all of life. Your peptide, your protein molecules, which are made up of short peptides, or your nucleic acid molecules, which are stretches of monomeric nucleotides strung together, are all polymers. So how do you go from this monomeric to polymeric space? And how do you then form minimal living assemblies is something that still beats us. We understand partial steps of the processes, but we don't still understand all of it. And this is why one of the interesting ways of trying to understand how life might have come about from the fundamental molecules is to basically ask, what is the least common denominator of all life, right from a human to a single cell entity, to a plant, to a mushroom, to a catfish, to T-Rex? Basically, it's cells. So if you can use these fundamental component molecules and concoct cell-like entities in the lab in a test tube, you can say that you have synthesized life using a bottom-up approach. Here is a slide which shows you the fundamental unit of life as we appreciate today. One is a prokaryotic cell and one The other one here on the right is a eukaryotic cell, which is what you and I are made of. What is immediately apparent is it's already very complex. Even the simpler of these two, which is the prokaryotic cell, which is your bacteria, basically, they themselves are super complex. So the minimal life that I'm talking to you about, please appreciate, is even more simpler than this. And despite being unicellular, it's just mind-blowing how these bacteria have occupied every single niche plausibly thinkable on Earth. Every single extreme niche. Here are 4 extreme niches that I'm showing you. One is hyperthermophile region where temperatures are really high. The other area which you will actually be visiting probably in the strip is a hypersaline lake where salt content is so high that you and I can shrivel and then, wither away. Then you have permafrost dwelling organisms, which really like the cold, like the very cold temperatures in Ladakh means some areas are permanently ice ridden. And here are extremely UV resistant organisms because way high up in such a high altitude sort of desert like environment, your UV that's coming in is really, really high, which is why you need to slather yourself with a lot of, UV resistant creams and stuff. So in all these extreme environments where you and I will struggle to live or actually even die, these single-celled organisms can thrive. So it's super important to first understand how did the ancestors of the single-cell entities come about. Here is a very cartoonist, minimal version of a tree of life, which shows that at the base of all life as we understand today, there possibly was a last universal common ancestor. This was the understanding till about a couple of decades ago. Now it looks more like this, where the appreciation is that you had these minimal living entities, a common ancestral community of primordial cells that interacted with each other, and some set of selection pressures, pH, temperature, salinity, availability of ions, availability of different kinds of molecules, all of this can shape what might have eventually survived and led to what we see today. So protocells, or these primordial cells, fundamentally are a bilayer constrained system, like a tennis ball. Take a tennis ball and cut it open, and that's what you're seeing here. Your bilayer is the cell membrane. Inside it has a genetic material of some sort, like an RNA or a DNA, and it's thought to have been powered by some minimal metabolic network. And it's important for us to understand that using just a laboratory-based approach to understand how this assembly came about is not at all helpful. You also need to go out to analog environments to better understand how the early Earth geological settings, which are still sort of encapsulated in some sense in analog environments, and the extremophiles that thrive there, that their biology, what might that tell us more about how can we think about these assembly processes? So to go back to the environments that are very relevant to this expedition that you're on, AMAS 2024, on the right side is a terrestrial geothermal environment, like us readily present where you are right now. Unfortunately, you might not be able to see such sexy looking hot springs, but having said that, these are the kinds of hot springs that are thought to have facilitated terrestrial geothermal processes relevant to life's emergence. The other environment is a volcanic system again, a geothermal system again, but present on the sea floor. And I'm not going to be talking about this for the rest of the talk. I'm going to focus on this environment here, which is the kind of analog environment which we are very interested in the lab to try and simulate these conditions. In the lab, we can use some approaches to minimally sort of simulate it, but unless and until we go to Ladakh, which is a Mars analog, and bring back hot spring water samples, you're remotely not creating the complexity that can be encompassed in these environments, which are crucial to better understand the processes that you're interested in understanding. How did the monomer of a nucleotide become a polymer of nucleic acid? How did the monomer of an amino acid become a polymer called a peptide or a protein? And these processes, instead of oversimplifying them, which is an important first step to understand the biochemistry underpinning these processes, it's also very important to understand what might these reactions look like in environments which are much more realistic, which are much more chemically complex. So in this context, terrestrial geothermal springs have been a real this is something that didn't gain traction for about 2 decades now. In the last 2 decades, it's become something that people are more interested in, and particularly in the last, I'll say, decade, it's become much more important. They include hot springs, geysers, like the kind that's shown here. If you can appreciate this image here, this particular hot spring has extremely gorgeous colors at the edges. And you know what that is? That is life. Life is thriving at these extreme environments. Though when we try and understand how do our processes interesting to us pan out in these environments, we are looking for more non-life hot springs. Hot springs that look very bland, very boring, but they're very interesting to us because they are very geochemically relevant. And those are the kinds of water samples where we only want to encapsulate the ionic species coming from rock water interactions. We don't care about life at this point as complex as a bacteria. We care only about those kinds of water samples which are emitted in these geysers, in these hot springs, which are rich in minerals, and inorganic gases present there actually impinge on the chemistry that is plausible there, and hence has a direct implication for the kind of ions that you can find there, the cations, the anions, so on and so forth. And these environments are very interesting because they provide very interesting chemical fluxes, which essentially allows for interesting precursor molecules to be available there. They provide very interesting energy fluxes. You can have day-night cycle-driven temperature differences there. You can have, if you think of a geyser at the edge of a tidal pool or something, or a tidal pool itself, at these high temperature conditions, at these high altitude conditions, day-night extreme temperature variations can also facilitate fluctuating temperatures. You have very interesting surfaces present around the water bodies here, which allow for molecules to attach themselves and undergo even more interesting reactions. So all of these reasons are why hot springs or terrestrial geothermal springs per se are very, very interesting analog environments for us to study in the lab and for us to sort of simulate in the lab to better understand the reactions that we are interested in. So we have a model for how we think life might have emerged in these environments, given the right fundamental precursor molecules. For example, the compartment forming molecule, the information oligomer forming molecule, and the catalytic polymer forming molecule. You can imagine how under the wet-dry cycle conditions or the rehydration conditions that very readily are driven by day-night cycles, by rainfall, by seasonal variations, you can actually have scenarios where when there is dehydration, you're pulling a water molecule out, and that is crucial for an oligomer to form. In your cells and my cells, your mononucleotides are stitched together via a phosphodiester bond to form a polymer using highly precise enzymes. But on the early Earth, you have a chicken and egg problem. Did the information molecule come first? Did the catalytic molecule come first? Because the catalytic molecule needs to be made using the information in the nucleic acid, so on and so forth. So complicated. So there is a very interesting hypothesis that provides some respite from this, and I'll get to it in a minute. But if you imagine that one molecule did both the roles, the idea then is to understand how can you form the polymer of that molecule, and how can you have it readily encapsulated during a rehydration stage in these membrane-forming molecules, which can now start looking a little like your primordial cell. I mean, look at this panel. Isn't that a bilayered membrane encapsulating a polymer, which looks a lot like that tennis ball thing that I showed you? I mean, it's not looking exactly like its cousin, but you get the drift, that you can actually form these kinds of entities just by starting with fundamental precursor molecules and simulating earlier geological conditions. And these inset images that you see here, this is a freeze fracture image of multiple lipid layers which have encapsulated these red balls. You can't see them here because they are diffraction limited. And in this panel, you're actually seeing similar kinds of multilamellar sort of lipid layers. As soon as they touch water, these molecules have a water-loving and a water-hating group. The water-hating group just doesn't want to interact with the water present in the environment. So it just balls up into these higher order structures called membranes. They could be single-layered, bilayered, so on and so forth. We are predominantly interested in what looks like our cell membrane, bilayered membranes, and they very readily form under these conditions. And as they grow out and bleb into these balls, they encapsulate the growing oligomer within them. So given this model for the terrestrial origins of life in geothermal pools, we try and actually bring back water samples from analog environments to understand how these reactions pan out in the lab. And we have a whole bunch of steps that we think are crucial for the transition from your prebiotic soup of molecules to what we think are primordial cells. I'm not going to get into the details of it, but suffice to say that if you have a whole bunch of these molecules shown here from 1 to 7, these are all the primary entities that make up your cell membranes. Your cell membranes are a little more complex in the sense that they have one head and two tails. Two tails that hate water, one head that loves water. But at the core of it, if you zoom in, they are made up of basically what are called fatty acid molecules, represented here in one. And very early on, when you didn't have very complex molecules present, it is thought that just these fatty acid molecules and their cousins, their derivatives, where the head groups could have been any kind of head group, could have had a phosphate, could have had a sulfate, ammonia, hydroxyl, alcohol. This is essentially an alcohol group. It could have been any number of these different head groups. At the right pH, they can actually form these pseudo double chain structures, which are just like what is there in your cells and my cells, and they can very readily assemble into these membranes. I'm not going to get into the chemistry, but I just want to talk about this one project just to give a flavor of how these vesicular structures, these bilayered vesicles, when you do them in lab simulated conditions, the experiments, they give you very different results. But the second you go out into an analog environment, the results are very different. And this is a project that was led by my grad student. He looks too young here. He's now a faculty at Indian Institute of Chemical Technology in Hyderabad. His name is Manish Joshi. It was led by him and very ably assisted by Anupam, my postdoc, and another IPHD rotation student who had gone through our lab. What Manish wanted to understand is precisely this. In analog environment, when you have a complexity in the precursor molecular space, how does the crosstalk between the catalytic molecule, monomer, and the membrane-forming monomer, what kind of outcomes might come about in these conditions? And that is the gist of the slide. You don't need to understand too much more, but he wanted to understand how increasing the molecular complexity of the starting reaction mixture in like what might have been present in an analog environment hot spring, how will that affect the crosstalk between the membrane and the monomer, which results in the catalytic moiety, basically amino acid based entities. If you can stitch them together, these balls of red strings like a red pearl string, these are actually short peptides. So they can, in principle, readily form during the dehydration stage. But if they are present in the presence of your membrane forming monomers, suddenly things start happening differently. But that's beyond the point now. What I want to show you is that in 2016, we for the first time went out to Ladakh to explore it as an analog site. We did it under the aegis of the NASA Spaceward Bound program. And three important hot springs were our focus. You're seeing them here. If you can see, you don't have all those gorgeous looking colors that you saw in the slide a few minutes ago, right? Here, it looks very boring. It just is a very geologically interesting setup for us because there's not much biology happening here, but this is precisely the kind of water samples that we want. And we explored three different water samples, one from Chumatang, one from Puga, and one from Panamik. And the pH these are certain physicochemical properties of these environments which are important for us to know before we start doing our reactions. What was the temperature? What is the pH there? And we also need to understand what is the ionic makeup of all these molecules, which is very difficult for us to do on site. So we bring back the water samples and we study here. But we basically brought back these water samples to study the following. If you have oleic acid, which is a fatty acid, or combinations of oleic acids and its derivative molecules with different head groups, how readily will they assemble into vesicular structures or those compartment structures under realistic analog environment sample conditions? So we had a control, which is what we do in the lab here. We use Bicene buffer, which is a goods buffer. It's at pH 8.5, I'm sorry. And when you use it, you get beautiful vesicles. We wanted to see if we can get the same kind of beautiful vesicles in these analog environment samples too. And here are pictures of the team that actually collected the sample. We had a very interesting mix of scientists from NASA, from Birbal Sahni Institute of Paleosciences, IISER Pune, and a few other parts from a few other parts in the country, and also from UK. It was a really fun group of people. And we explored these three hot spring sites for various, we sort of systematically characterize the various parameters on site. Here are hardcore geologists, Rakesh partly maybe, but This gentleman here is a full-fledged geologist, so he had all these very cool gadgets that he brought along. What we did is this is my first student, Chaitanya Mungi, who graduated about 5 years ago now, and he was crucial in collecting the samples, along with another grad student of mine whom I can't see here. So we brought back a lot of these samples to the lab, and we first did geochemical analysis of them. And I won't get into details, but this is just to say that if you have Puga sample, the spread of major cations and anions, the amounts present in them is very different from Chumatang and from Panamic. Is that something you can appreciate from this table? You can just look at the columns and look at the numbers. Your sodium ions, for example, is highest in Puga, followed by Chumatang and then Panamic. If you take magnesium, Puga has no magnesium. It has very, very little amounts of magnesium. There is very, very little amounts of magnesium present in Chumatang and Panamic. Now, if you look at the anions, for example, if you look at chloride, Puga is rich in chloride ions, but very little of it is present in Chumatang and Panamic. What we wanted to know is if the variation in these ionic species had some complications or implications, rather not complications. We later on learned that there are complications, but we wanted to know if there are any implications for them to form these bilayered vesicular structures. And we don't need to understand the remaining, but suffice to say that some did much better than the other. In this table, you're looking at the fatty acid system. Chain lengths are varying here, so you don't need to understand beyond a point. One is a 10 carbon chain length, one is 11 carbon chain length, one is a 18 carbon chain length. Oleic acid is what I'm showing here, along with its two derivatives, oleal alcohol and glycerol monoester containing head group of oleic acid. And if you look at it, the one which has both the oleic acid and the glyceride, the vesicle made up of these two components, did very well in all of the hot spring samples, whereas it didn't do as well. The others didn't do as well in all the combinations. Particularly your Bising buffer, everything in the control conditions, everything formed beautiful vesicles. So just by looking at that, you can't go, yes, vesicles form ho gaye. But the second you step into the realm of using analog water samples, the story is very different. So what analog research primarily tells us is that you need to interpret your lab simulated experiments very carefully. You cannot just assume that what you learn from that is all. You need to challenge yourself to actually complexify the starting reaction mix by actually working with these real spring samples, spring water samples, to better understand what truly might have been the survivor or the victor under those different complex conditions. This is just to show you two images. Here on the left side are all the four systems: oleic acid alone, oleic acid with alcohol head group derivative, oleic acid with the glycerol monoester derivative, which is the one which formed vesicles in every condition, under every hot spring condition. And here is when you mix all three of them: oleic acid, oleal alcohol, and glycerol monoester. Under buffered conditions, every single thing is showing you these ball-like structures, right? Shown by white arrows, they are vesicles. But the second you move to hot spring samples, and here is an example of Puga hot spring, you suddenly see only the oleic acid and glyceride forming the ball-like structures, like in this case. And in the other three cases, you're forming some random things. I won't get into the chemical nature of these identities of these things, but these are not vesicles, and that is confirmed. There are many biophysical ways in which we can determine that. So hopefully the story has left you with this understanding of why analog hot spring samples are super important for us to let me try and get out of this book. Yeah, why analog hot spring samples are super important for us to better understand, reactions that we're interested in. There's a whole bunch of other things I can tell you, but for now, suffice to say that while you're there, kindly appreciate the fact that it's extremely important for you to appreciate a whole bunch of geological processes that impinges on the chemistry that's possible in these niches. It could be any kind of niche that you're interested in. And then you take that information back to the lab, or you actually can sometimes do experiments right then and there, but it's very complex in terms of following through with the analysis, because you can't carry giant instruments with you, which allow us to do, that particular set of images that I showed you comes from extremely high-end microscopes, which are this huge. You cannot carry them and go. And by the time I can fly them to Ladakh, they'll stop functioning. They cannot deal with such kind of travel and everything. You can't even take a light microscope and image them, because these are very extremely small vesicles. I didn't talk about the scale bar, but they are literally 400 nanometers, very, very small, half a micron length. Yeah, half of a bacteria. They are half the size of a bacteria, pretty much. So it's very hard to do some of these high-end experiments there. So you actually work with, you do the first set of initial preliminary analysis there. You bring back the samples to the lab and you try and study here. But if you take examples like the one that Renita is interested in, she's very interested in extremophiles. She's also going to be collecting some extremophile related samples, possibly. She also cannot sit and characterize them out there in the environment, right there at the site. The environment is great and awesome. And if there was a university nearby which had access to all these instruments, we could have sat there for 3 months and figured things out, but that's not possible. So you bring back samples very, very carefully back to your institute, wherever you're working at, or collaborate with a person who has access to this and who understands how to acquire this data and appreciate it. And then you basically make interpretations in a much, much more systematic and realistic manner to better understand how life emerged on the early Earth. Thank you for your attention. I'm happy to take questions. Here is just a slide leaving you with some of the fun things we do when we are not doing science. Renita?
Yes. Thank you so much, Sudha. Anyone, any questions?
Let me stop sharing. So it's sad. Oh, I can see you people suddenly. Yeah. I went a little fast there, but the idea is take home message. All that you need to remember, whatever you wish to appreciate about processes that happened really far back in time, where you don't have too much idea about all the specifics, you can actually go out to analog environments where some of this complexity, of course, you're not looking at a 4 billion year old sample. Please appreciate that. You're looking at a sample that is modern, that is current. But having said that, those environments are extreme enough in terms of temperature and pH that the molecules that can be formed in those environments are still probably reminiscent of what might have been very early on. So you encapsulate that complexity in a tube. Aditi probably will be collecting some samples for us if Renita and Pavitra permit it. So when she comes back, the hope is that she will do her master's thesis with us, where she is going to try to understand one other very important problem in our area, which is to understand how did the informational molecule come about. So yeah, you're there for a reason. In addition to origins of life research, analog research is super important for understanding paleoclimates, has fundamental ramifications for climate change related processes, so on and so forth. So others who are going to be joining you either online, or Renita and Pavitra, and I think Vinod is also there, will be talking to you about some of these implications while you're sort of spending your time over in those gorgeous locations over the next week or so. So yeah, how many of you are science students? Can I see a show of hands?
I mean, all are science students, but we have geoscience and
Renita, choppy. I didn't, after geoscience, I didn't. Renita. I'm cutting.
Yes. Basically, we have 4 of them. We have around 4 of them. Hello.
I cut my video off just so we are on the same.
Hello.
I can hear you now. Can you hear me? Hello.
So we have geo-bio. Can you hear us?
Now I can hear you. I lost you after geoscience very early on.
Yes, geosciences, biotechnology, and astrobiology students.
Of course, then you all should have understood what I said better. But somehow I didn't see that register on your faces. Either you're tired or it's the lack of oxygen. I don't know what it is, but please make sure you're breathing consciously, and please make sure you're taking care of yourselves. Ladakh is not a very, forgiving terrain. So you have to be very, very careful. You people are still in Leh, right?
Yes.
Okay, when do you head out? Tomorrow?
Tomorrow.
Good luck. Good luck. Have fun. Enjoy. But I'm happy to take questions. If you're feeling too shy or too tired to ask them now, you're always welcome to send me a mail after you come back from the expedition.
Anyone, any questions? Okay. Yes. Aditi, you have a you have questions? No. Okay.
Okay, then I'll say no questions. Then I'll just once look at all of you and say bye.
Yes, we have no questions.
क्या बोला? I didn't understand. What did you say?
All right. Yes. Bye bye.
Bye.
I
Have
said
fun.
bye.
Bye, Pavitra. Bye, Navita. Bye, Aditi, Amar, and everybody else. Bye, bye, bye. Ciao, ciao. Bye, 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 bye. See ya. Bye.
Bye bye.
Bye, ma. Bye. Thank you. मुझे
Should I close the meeting, ma'am?
Yeah, thank you so much. Thank you. I'm leaving.
Thank you. Thank you.
Munazir, you just keep it logged in. 6 o'clock, we have one second. Hello? Can't hear you. You're on mute.
Now you can hear me?
Yes. Hi, Bharti. Hi, Bharti.
Hello, ma'am. How are you?
Good, how are you?
I'm good. I'm good. The
You look
banner
great.
looks. Thank you. The banner looks great.
Yes.
The banner looks
But
great.
amazed.
Amazed.
Over there, amazed.
Yeah.
You look very corporate like.
I have to look like a founder. Then scientist who's a founder.
Yes, yes, I agree.
Thank you.
Just before you start, heads up, the internet bad. So if we lose you or something, I'll send you a message or I'll call you on WhatsApp so that, if, do you have slides or are you just like doing a impromptu kind of thing? Slides?
So I thought that I'll make presentations, but then because of the merchandise issue, I realized, okay, it's better to go just with the flow. So I'm not presenting anything. And I think with the bad internet, it's
Okay.
better to speak up only.
Okay, then that's fine. So you can just tell them about, whatever you want with Ladakh, your company, and how it helps in the region, and also a little bit about Geo. We have quite a few Geo people this time,
Okay.
so maybe they'd be more excited to hear from you. We'll see.
Yeah, I was thinking to give some food for thought questions for the field trip. Like, we found sulfur on Mars. How is it relatable with the astrobiology thing? And is there anywhere we can find sulfur in Puga or Sokar and permafrost? That's just the basic questions I'm gonna give. Other than I'm talking about paleo lake sediments and stuff.
I think we can start. So we have 15 participants. A few of them from our MSc Astrobiology program, from our biotechnology program. We also have geoscience participants, 2 participants from the National Remote Sensing Center, ISRO. They are also from the geo background.
Wow.
So I think we are good to go.
Okay.
Yes. Yes.
Great. Hi, everyone. So I'm Bharti Sharma, geologist and founder of Adya Planetary and Geoscience Research. Am I audible, right?
Yes, you are.
Okay, great then. So I'm an analog astronaut as well and a geologist. So a little about my background and about my company. So why I started Adya, it was because of Ladakh. I started my planetary geology journey in Ladakh in 2021, where I was serving as a field teaching assistant and research assistant with MIT Mumbai. So I was assisting senior scientists like Benita Fathial ma'am, Jonathan Clark. So I was assisting them in their geology project, atmospheric science project, and climate science project. Because of my background in earth science. So I have studied all the domains of earth science, atmospheric, planetary, climate, geology, everything. So that became an edge for me. And gratefully, I did my bachelor's from MIT University, Haryana. So there was a MIT connection, and then I was exposed to astrobiology there. So I found this field, geobiology, very relatable. I was like, okay, wow, that is so intriguing for me, because as a geologist, I used to look at the rocks, the soil, the landscape, everything. But when I started volunteering with the astrobiology team of MIT Mumbai, I realized, okay, there are microorganisms, there are bacteria which can be relatable to geology part, how these things evolve with the geology. So that field was intriguing, and I volunteered with N number of projects during those 2 years. And I realized, okay, there is a great chance for a geologist to be in the field of space exploration. Then I started volunteering with space startups. And that field is honestly in India or elsewhere is dominated by engineers. So I found myself as a geologist or a science person working with thousands of engineers. And they used to build rovers. So I have done project work on rovers and on CubeSat satellites. And they need the expertise of a geologist to know why they are going there, what is the science behind those technology. So I realized, okay, there is a gap for the collaboration for the science people. So why not make something for all the science people, for all the geologists or biologists? So I started Adya Planetary and Geoscience Research. So one domain of Adya deals in the collaborative research part. So when I was working in Ladakh and when I was volunteering with these space startups, there are n number of independent scientists from everywhere in the world. There are a few from India, but mostly from other countries. So I want to inculcate this behavior or this project of independent scientist in India. So as my startup like Adya, you can take this as a secondary affiliation, or you can be a part of this project. So for instance, you are working as a remote sensing expert or as a geographer, and you don't have the exposure of a planetary science or planetary geology for say, or astrobiology. You can come up to Adya, you can work on the ongoing projects, and you can be a part of a culture where the collaboration works, where anyone from different backgrounds can come up, of any age, of any background, of any country, can come up and do these projects. So that's what first domain of Adya works in, and that's how I was inspired by Ladakh. My second domain is geotourism and geoheritage conservation, again inspired by Ladakh. So when I was doing my fieldwork in Ladakh, I realized, okay, there are so many things which are, just there. So when you go to Stakna, which is one of your field site as well, you will see the rock paintings, the petroglyphs over there. And that is just one part of petroglyphs. When you roam around Ladakh, you'll just get on the roadside and you'll see the petroglyphs. That is just there. And nobody is protecting it. And it's so abundant in Ladakh. You'll realize, okay, the conservation part is of utmost importance in Ladakh. So when you'll go to Sokar, when you'll go to Puga, all these geoheritage sites are in GSI as well. That is Geological Survey of India. It is marked in GSI, but are they being protected by the locals? So when we were teaching students, and there were some localites with us as well, whom we were teaching about the geology of Ladakh, they were also clueless about the vast geology background of Ladakh. And I realized, okay, there is a need for community teaching. Like, people know science, but do they connect with it? But they do the importance of knowing the geology. So my current project in Ladakh is about geoheritage conservation. So I am doing field work in Ladakh. I recently completed my field work in Ladakh, and we were teaching communities about the geology of Ladakh, like how geology, how Ladakh was evolved during the geological time scale, why it is important to conserve each and every site. The Ladakh is called the Shangri-La, where, find these hidden valleys, which is connected to limestone, So how this mythological stories they know, but they don't know the science behind it. So you have to connect with the science and with the mythology, with their culture in such a polite and, great manner, so that they want to learn. Because whatever we are doing in science exploration or space exploration, it is incomplete without the community involvement. So that's my job, to connect science with the community. And that's what I'm doing in Ladakh and other parts of India, as a geoheritage conservator and as a planetary geologist. So all of you are here in Ladakh for the planetary geology part. Do you know why Ladakh is a planetary analog for Mars? Can I get any answers? Like, if it is okay.
Anyone would like to answer?
Do you know any similarities of Ladakh and Mars?
There are many similarities between Mars and Ladakh surface in terms of geomorphology, in terms of terrain, in terms of extreme environment.
Can you name any one similarity, like anyone?
In terms of rock type, sedimentary rock?
Rock type, or maybe you said geomorphology, like what landscape feature you find here in Ladakh and on Mars as well?
We have drainage system pattern which is similar to Mars. We have hot springs. So these are many features which is similar to Martian terrain.
Yeah, that's lovely. So one of the main feature is actually the gully system. So gully system is the glacier gullies, which you will find in Sokar. When you will go to the other side of Sokar, you will see, even if you don't go to that field area, you will pretty much see the gully system in Sokar and Buga region as well. So they are very deep. They are formed because of the glacier meltwater. And from there, they feed the main lake system, either Puga or not Puga, Pangong, so Sokar, Somorari, so like this. So this gully system is quite evident on Mars through the remote sensing data we have gathered. And apart from gully system, the hypersaline lake environment is quite evident as well, because we have find salt crystals and permafrost everywhere. That is the remote sensing data suggest. So because of these similarities and because of the cold desertic environment in Ladakh, Mars and Ladakh are quite similar. And as per the astrobiology is concerned, Renita ma'am is the best person, because I have worked with her and I know how deliberately or how, with dedication she works, and she relates with geology and biology a lot. So because of her, I got to know that, you can find these lichens and other bacteria and microorganisms or heliophiles, which there is a good chance that you will find it on Mars as well. So because of this geological similarity, we go next step into astrobiology. Okay, if we are finding these bacteria in this environment, is there any chance that we will find this heliophiles or other bacteria on Mars as well? So that's how you relate geology and biology and Ladakh and Mars. And apart from hypersaline lake or hot springs or cold desertic environment or the gully system, you'll find the paleo lake deposits. So when you are going towards Sokar or when you are going towards Puga, you'll find many paleo lake deposits. So you have to differentiate between if that is a paleo lake system, or if that is a proper succession of some rock system or formation. So when you will find the paleo lake system, so when you are going towards Sokar from Leh towards the airport or from the airport, you'll find there are different small layers of soil. So when you are going to sand rams as well, that is nothing but paleo lake deposits. So a great example in Ladakh for paleo lake deposit is Lama Yuru. So that whole Lama Yuru place was once a lake bed of an ancient lake, and they have a mythology related to it, that there was a time that there were some angels and there was no water in that region of Lama Yuru. And so the angel gifted the people of Ladakh that, there will be some water in the area. I'll just, do some meditation and things. So he performed a ritual for that, and that Lama Yuru was flooded with water. So that's how Lama Yuru was formed. And then after a point, like after 100 or 200 years, that's their folklore. So after 100 or 200 years, they realized, okay, now we don't need water, or I don't know why they said, because they were either migrating from Lama Yuru to other place, and they want to find some gems, which is close to Vanla as well. So in Vanla, you will find really good herbs, which they use for local medicines. So they wanted to extract the water from Lama Yuru. So the saint again did some ritual and some practices, and he extracted the water from there. So that's how the lake bed is quite evident there. So what similarities you will find in Lama Yuru is quite similar to the region when you are going towards Sokar, not in Sokar, but when you are going towards Sokar. So that this paleo lake environment is also quite evident on Mars as well. Now, I would like to give you some food for thought questions that you can, relate or find a place on Mars where you will find the paleo lake system. And secondly, why Ladakh has hypersaline lakes? Is it related to planetary geology, or is it related to altitude? So that's the question for you. If you want to answer it right now, that would be great. But by the end of the field, you will get your answers, that why Ladakh has hypersaline environment, and is it related to planetary geology? If yes, then why? And how is it related to Mars then? I am telling you, I'm giving you a hint in advance that it is related to the altitude of Ladakh, the elevation of Ladakh, and secondly, then to Mars as well. So you have to find the dots between them. That's all from my side, from my current work and from my company and what I know from Ladakh. If you have any questions related to geology of Ladakh, do ask me.
Okay. Thank you, Bharti. Does anyone have any questions about the geology of Ladakh before we head out to the field? Because Bharti has a lot of experience about different locations. So if there's something you'd like to know, or some structures or features you'd like to see, or anything like that?
Hi, Bharti. This is
Hi.
Prabjot. All the points that ma'am has planned, we will able to see any eclogytes exposures there, eclogytes or chromites.
So you will see the eclogite when you are coming. So are you guys coming from Sumdo region? Like, are you guys planning to come back from the same route, or you are planning to get a diversion from Sumdo?
So we are coming from Sumdo.
Yes, you will see the eclogites there.
Can you pinpoint that location, because I want some samples of chromites and eclogytes? If you have the
So
location.
I can forward you the GPS locations, and I'll forward it to Pavitra ma'am. You can get the locations. But the region is after you cross Sumdo, when you are coming towards Chumathang, in between that region, you will find the eclogites.
Okay, thank you so much.
Welcome.
I will be needing your contact today. So.
Yeah, sure. I'll forward it to Pavitra ma'am.
Yes, anyone, any more questions? I just have a general question, Bharti. As a geologist, what was the most difficult, like, what was the biggest challenge you faced at the Ladakh region? Like, it can be related to sample collection or identifying features, or is there something specific as a geologist you found challenging?
So there'll be three things I'll mention. First is the acclimatization. So if you're not acclimatized, because that was the problem what I faced, if you're not acclimatized, you will find difficulty in collecting samples or doing the GPS things or doing the GPR things. So that sample collection and doing fieldwork is quite hard when you are not acclimatized properly. Even if you are acclimatized, you have to go very slow. And usually in geology field trips, we have to go very fast because we have to collect thousands of samples and cover n number of places. So that was quite a challenge when I was there first time. So that's my advice for all of you, that be slow all the time. The boring the field is, the most useful or the most fruitful the field is. So you have to be boring. You have to be very slow when you are collecting samples, when you are hiking up, or when you are roaming around the spots. So you have to be very slow because acclimatization needs time, and Ladakh is not a place for fun. So it's quite a hectic thing for us who are research scientists who are working there, not going for fun events. Secondly, with the rock type, yes. So the geology of Ladakh is quite active and quite tectonically influenced. So once you are seeing the shale rock system, and you just cross the border and you'll see the sandstone as well. That's okay when you are in the lower Himalayas or other regions of India or other parts of the world. But in Ladakh, it's quite complex, because one layer is shale, the other layer is sandstone, the other layer is shale again, and then there's limestone again. And because these mountains are very much folded and jointed and faulted, you won't see a proper formation. So if you want to analyze the proper formation of that particular region, you have to hike up. You have to see at the bigger picture, you have to hike up, and you have to look at different layers, and then things will be more clear. So when you are going through the walk or through the highway, you will see different layers coming together. So you have to understand that this was once a horizontal bed, and then it was tilted and faulted up, and now it's a vertical bed, or either it's a 45-degree angle tilted bed. So you have to understand that kind of formation and that kind of tilting. Then only you'll able to analyze, okay, this was the formation first, and that was the second, and that was the third. So when you are going towards Sokar, you'll see the sedimentary and metasedimentary rocks. So you have to know that sedimentary was the basin, and then the metasedimentary rocks were there. So, and once you go towards Buga, you will find the metamorphics as well. So you have to understand this complexity, and that's why you have to be very slow, because the formations or the rock system change so frequently. And then if you have done the field work there, you know that it's very hard to analyze everything when you are on the highway road. You have to hike up to understand the whole geology. Otherwise, you have to be very open in mind that, okay, this was once a horizontal bed and then it was tilted. So the colorful bands and all was once a horizontal bed. So once it was sandstone, and then it was limestone, and then it was shale. So, and then it was tilted, or they are vertical beds. So the route from from Ukshi till Sokar is all sedimentary rocks. And that's such a beautiful, colorful rocks. And those are all tilted rocks. You'll find really great samples over there, really good formations, and you'll see the tectonic activity, or the impact of tectonism on Ladakh there only, because you'll see really cool vertical beds over there. So those are the two things I find quite difficult and quite challenging, but also very fun to do. Apart from the acclimatization, yes, you have to be very slow. You have to be mindful of your own health. But this formation thing, the complexity, you find it challenging when you are dealing with it, when you have to be, thorough with what formation is this or what rock type is this. But it is quite fun as well, because we used to be frustrated sometimes that we don't know what rock type is because it is so much fractured, and the bedding looks like, okay, this is a proper hard rock, but once you take the sample, it is all sand. And then you have to be like, okay, this was highly jointed, highly fractured area, so it could be a fault zone, it could be because of the erosion. So that complexity you have to keep in your mind.
Okay, thank you. We have a few more questions.
Yeah.
Go ahead. Come up.
Ma'am, you said about hi ma'am,
Hi.
And I'm a student of MSc Astrobiology in MET. So, ma'am, my question is, you mentioned about the high elevations and the thing just to slow down, but if like in Tanglangla, that is really very high. So, if we find ourselves in some kind of uneasiness or work, so what would be your advice for us to do?
So when you are going to Tanglangla, don't get out of the bus for more than 30 seconds or 1 minute, not more than that. Because last time we faced this problem, students were out for like 3 minutes, 5 minutes, 7 minutes, and they were facing some real issues. So you have to be mindful of your own health. You have to get out, take the picture, and come back to the bus. Okay, not more than 1 minute. That is the maximum. 30 seconds is the ideal what we felt, because, just get out, take the photo, and come back. If you feel little dizziness or little bit of unconsciousness or something like that, keep sipping water. Okay, have your water bottle with you, keep sipping water, and get those orange candies. That helps a lot. So what we used to do, once we are in Ukshi, we'll buy those candies. Until Tanglangla, we used to have those candies in our mouth, and that helps a lot with the acclimatization part. I don't know, it's because of the mind or it's the toffee working, but it actually worked for all of us. And don't get out in Tanglangla. Also in Sokar, once you are there, you will be reaching there by late afternoon. So get rest, get properly rested. Don't venture out in the night. It looks beautiful, but don't venture out because you have to acclimatize. So you have to sleep there to get acclimatized. So take proper sleep. You won't get more than 5 hours or 4 hours of sleep, because that's how our mind works over there. But you have to be in the bed for at least 6-7 hours so that you are properly acclimatized. You have to spend the night properly. Don't venture out without telling your superiors. But yeah, keep those orange candies, keep your water bottle ready, and don't venture out. That's the 3 advice I would like to give. And be slow all the time.
So that Tanglangla pass is it's just like a peak, and then you start heading down. So that is why it's only like a 30-second stop. So you don't have to get over the bus.
It's just for the photos, actually.
That is what, even in the bus or wherever, we cannot stop there for much longer period of time. Once we are there, it's like a peak, and then you start, you start coming down. So it's just like that one point, very short point, and then you start. You may find many tourists, standing, clicking pictures, all that, but then it is not advisable, because after that we have to go again to a different location. Right? Okay. Yeah. Yes. Hi, ma'am. I'm Myra from Ferguson College. So
it's kind of a basic question, but you mentioned that there are a lot of different rock systems here. So what exactly is the reason that there is so much variety in the north?
Because it was different plates. So when you are going towards Sokars, you'll see one Trishul point. Okay, so there is one huge Trishul on the top of the mountain. It's guarded by the army area, but used to make a stop over there. So you'll see you are on the actual line of contact, where on the left hand side is the Indian plate and on the right hand side is the Eurasian plate. So this Eurasian plate was already there in the north side, and then the Indian plate was cut from Gondwana land and started its journey towards the northward, and then it was collided with the Eurasian plate. So there are two continental plates. One is Eurasian, second is Indian plate. Then in the middle, when there was no collision, there was Tethys Sea. So that Tethys Sea bed are also there. So if, the variety of rocks and variety of formation is because of these three different plate system. One is the Eurasian plate, the other one is the Tethys Sea, the other one is the Indian plate. So when we are in Ladakh, we are actually beyond the Himalayan region, that is the STZ zone. So you will find mostly the Tethys bed here, the sedimentary rocks which belong to Tethys bed here. And then there is a chance that you will find, if you are in the southward, you will find some rock system from the Indian plate as well. And then we have the Eurasian plate. So hairy. So you'll find igneous rock from the Eurasian side. So whatever igneous you are finding, it belongs to the Eurasian. Whatever sedimentary rocks you are finding, it belongs to Tethys bed and to the Indian plate as well. The meta rocks or the meta sedimentary rocks, the metamorphic rocks and meta sedimentary rocks, it is because these rocks, the upper surface of Tethys and Indian plate went through a lot of pressure and temperature, and then they change their chemical composition, and because of that you find these meta sedimentary or metamorphic rocks of Tethys bed and of the Indian plate as well. So that's because of these three plates and because of different actions and collisions and tectonism, you find different rock types and different formations. That's how it becomes quite difficult to understand, okay, this is there, that is there. So you have to be mindful, okay, this was all horizontal at one point of time. It started collision, and after that there was some upliftment as well, because when two plates are colliding, it will uplift. So because of the upliftment, you will see the bed at the top, and whatever was on the surface will at the top, and the lower part will be was at the bottom of the sea, and then it became tilted. So now the top part is at the lower end. So because of these tectonism and because of the active geology over there, you find different formations, and that's the complexity. And because of these complexities only, you find different rock colors. The bedding I'm talking about, the different colorful rocks you'll find along the Sokar route. It is all along the Indus River, and it's beautiful to see. And that's the reason behind it.
The second thing was not exactly a question, but I want to confirm if that's correct or not. The answer to your question, why hypersaline lakes are there in Ladakh. So I think it had something to do with the sediments from the glaciers settling into one lake, and then the sun kind of dehydrating the liquid and gets very concentrated.
Yeah, you are almost there, almost there. But you'll get the exact answer once you are back from the field. Yes, you are almost there, that the water gets evaporated and whatever is there left behind. And the reason behind it, because of the high altitude, the evaporation rate is quite faster. So that is similar to Mars. You have to answer that. And how is it similar to Mars? Is the altitude same or the temperature same? And what was the early history of Mars, the geological history of Mars, why this was the reason? So you'll find all this answer once you are there, and ma'am will explain the astrobiology part of it as well. But you are quite there, almost there.
Okay. Anyone, any more questions?
If you want to plan a geological field to collect different types of rock samples, what should be how many days we have to plan? And from your company side, you will be with us, or how you will support us?
Okay, so in my geology field trip, what I organize, I am always there. to assist. And I have had the experience of Ladakh and other parts of India. So I am your point of contact for everything. So like for Ladakh, you want to choose. So you have to tell me the section. So currently we are planning a field trip in south of Ladakh and in towards the Kargil side. So it's of 8 to 10 days of field trip. So usually if we are covering just one part of Ladakh, either the southern part, that is the Sokar part, you need 4 or 5 days. If we want to cover more towards the Kargil side, you add more 4 to 5 days. And then if you want to cover the northern region, that is the Neoma, Nubra and all, then you have to add 5-6 more days. So in general, it would be like 20-25 days field trip.
I'm interested there some pegmatites exposures are there, or any hydrothermal alteration.
Sorry, what? I didn't hear properly.
If any pegmatite sections are in that, or hydrothermal alteration mineral sections are there.
So in Nubra side, you'll find the pegmatites. Okay, and the best example for pegmatites is in the towards the Somarari section. So you'll find the pegmatites over there. And the hydrothermal vent system is in Puga, Chumathang there. So you want to cover some part of Nubra and some part of southern Ladakh, that is Sokar, Somarari, and towards the Hanle side. You'll find both the sections over there.
Thank you. Please be in contact. I will want to talk to you.
Sure, sure. I'll pass it on to ma'am and she'll pass it
Thank
on to you. Okay, thank you.
Okay, anyone? Any more questions? No? Okay, I have one, Bharti. So basically, we are just talking about astrobiology, geology, microbiology, and all this. But then you are now not just a geologist, you are an entrepreneur also.
Yeah.
So how was your journey, geologist to entrepreneurship? Specifically concerning, we have a lot of students that can also be one of their career options if they want to.
So
if you can just.
Yeah, I always wanted to be a scientist. That was my, childhood dream. I was inspired by Kalpana Chawla, and I wanted to make my career in space science. I wanted to be in the research field all the time. But after my master's, when I started volunteering with these space startups, it was the COVID time 2020, and I started volunteering with these space startups, Brahmastra, SSERD, SSERD, Brahmastra, and Blue Marble as well. So what I realized that, first, that it's a very collaborative research environment. And I like that idea because when you are working traditionally in India, you don't find that collaborative research environment when, you can see independent scientists coming in. And I love that thing about the engineers of India. They just come up with different topics and they'll be like, 5 guys sitting or 5 group of people from different fields sitting, and they'll just start doing their research. I like that idea. And what I realized, okay, geologists are missing a lot here. And after my master's, I volunteered with MIT Mumbai for the Ladakh project. And I wanted to continue with my PhD with everything. But then I realized, okay, there's a lot I can do as an entrepreneur than just being a scientist. So I can be a scientist, I can be an entrepreneur as well. So this thing was inspired by Blue Marble Space and with the work environment I got because of MIT Mumbai and Ladakh, that you can be both. So the idea I was inspired for the geology and geotourism was because of my mentor. So when I was in my bachelor's, he had a company called Litho Tours, and he used to organize these geology field trips for us. So he's a proper geologist, but he was into geotourism and geoheritage conservation. So he used to organize these things. And I was inspired by this. Okay, he's a geologist, he's working as an entrepreneur, and you have the ability to control your own decisions. You are your own boss, which has its own pros and cons. So, okay, so can I do this in India? Do I have all the, legal licenses and things? So thankfully, my brother and sister are lawyers. So I got the advice from them that if I want to start my own company and I want to be a scientist and an entrepreneur as well, can I do this? So we found out that legally, yes, you can. You can be, you can have your own research company, and because these the engineers I was working with, they were having their own company. So I thought, okay, why not? A geologist will have their own research company. That's such a new concept in India. And then I founded Adya, which literally means that the origin of everything, the initial reality. Adya, I took the name from one of the Hindu sacred texts, that is Srimad Bhagavad Puran, if I'm not wrong. So Adya translates to the initial reality, and it resonated with me that, with geology, I am talking about the origin of everything. And with planetary science, I also want to talk about origin of everything. So that name was, quite apt for me. And I founded this company, and I put two domains in this. First is the geotourism and geoheritage part, which my sir was already doing, and I kind of carry forward it with his guidance as well. He's the senior advisor in my company as well. And second part was collaborative research. So I got all the licenses. I got it through the MSME UDI thing portal, and that's how I started it. So I don't wanted to limit myself to just be a scientist. I wanted to explore more boundaries. I wanted to explore new collaborations with people. The PhD dream is still not over. I still want to pursue my PhD. I still want to be into the research in depth and maybe postdoc someday. But yeah, I realized, okay, this is the way I can give more back to the society, and I can bring more people into what I am thinking or what I'm believing will change something in space exploration or collaborative research. I hope I answered your question.
Thank you so much. Anyone, any more? Are you hiring, Bharti? Bharti, are you hiring?
Currently not, but maybe next year. But I'll surely contact everyone, whosoever wants to join the company.
Okay. Okay, anyone, any more questions? If no more questions, we shall end up over here. Tomorrow we are moving.
All the best to everyone. Keep it slow, and all the best for the Ladakh trip. Ladakh is such a great place. You'll fall in love with it. Trust me, I am already in.
Thank you, Bharti. Okay. Thank you so much, Bharti, for your
Thank you
time.
so much.
If there are any more questions or something, we will share your email ID, or they can also tell us the questions and we will share your contact.
Okay, sure. I'll forward all the links to you. Okay.
Okay.
Thank you. Thank you so much for having me. It was such a great seeing you again, and it feels so good. And all the best to all of you. Bye.
Thank you so much.
Thank you.
Okay. You can close the session.